Hello friends, in the earlier class we studied about the quality characteristics of food, that what are the various quality attributes, how do they change during different processing operations or during storage and handling operations, and ultimately how they influence the quality of the materials. Now, in this lecture of half an hour, we will see that what are the different factors actually which influence the quality of the food during processing and storage. And ultimately, these variables need to be controlled during processing and storage operations in order to get desired quality in the product. So, the important variables include temperature, time, rate, which is you can see here dt by dt, if the T is the temperature and small t is the time. Apart from these, the other components like pH of the material, pH of the ingredients which are used, composition of the product, composition of the gaseous phase, and last but not the least, water activity. These all factors become very important contributor to the overall quality of the product, and therefore, they should be meticulously controlled in order to get desired characteristics in the final product. We will now discuss one by one little details of all these factors, and to start with the first and important factor, temperature. And as I told you, it is important, in fact the most important of all the variables, because of its broad influence on almost all types of chemical reactions. It has very important influence on different processes which take place in the food, influencing its quality. The effect of temperature on an individual reaction rate has been well explained. It is generally expressed by Arrhenius relationship, which is K is equal to A e to the power minus E A by R T, where E A is the activation energy, R is the universal gas constant, and T is the temperature. The data which conform to this equation Arrhenius relationship, they generally yield a straight line if log of the reaction constant is plotted 1 by T, they give straight line, and this is well established as far as the chemical reaction kinetics is concerned. However, in the food processes, in the food reactions, it has been seen that the components of the food, they usually conform to the Arrhenius relationship over a certain intermediate temperature range only, and this we can see by taking an example of non-enzymatic reaction as well as an example of an enzymatic reaction, and see how the deviation from the linearity takes place during the process. In the non-enzymatic reaction, as you can see in this picture, that is here only during this range it is following the linearity, that is Arrhenius relationship. Deviation from the linearity is taking place over both the sides, whether you go on the higher temperature side or you come to the lower temperature side. On the higher temperature side, above a certain value of the temperature, depending upon the product characteristics or other variables present in the system in which the food is being processed, there might be deviation from the linearity, which you could see here in this figure indicated by A, that, and this deviation may be because of the change in the path of the reaction, mainly due to change in the concentration of the reactants. You can take an example of an evaporation process, where changing temperature above a certain level might result into the evaporation of the water, and this might cause the increase in the concentration of the solutes, and this change in the or increase in the concentration might result into the deviation of the process or deviation from the linearity. When the temperature is lowered below the freezing point of the system, with the progress of the time, ice phase, which is essentially pure, enlarges, and the fluid phase, which contains all the solute, diminishes, and the concentration of the solutes in the fluid phase increases, and this increase in the concentration of the solute in the unfrozen phase causes change in the properties of the components, and this change in the properties can decrease or can increase the reaction rate, which you can see here in the point B in this figure, and that of course depends upon what are the different components, nature of those components. Now, if we take example of enzymatic reaction, enzymes are generally protein, and the temperature has influence on the protein. So, in the enzymatic reaction, there is every enzyme has got some acceptability that over a certain temperature range, that is enzymes are stable. Going beyond that range, the enzyme reaction process is affected, that is above a certain temperature, the enzymes might get denatured, all right, and in fact the denaturation process competes with the product formation process, and there is a competition, and of course that is over the higher temperature side, there is the denaturation process dominates, and even the protein gets denatured, reaction might even stop, and that you could see here in this figure at point C, that is above a certain temperature the reaction rate is getting decreased. On the other hand, again here in this case in the enzymatic reaction also, that is in the vicinity of the freezing point of the water, subtle changes such as the change in the dissociation of an enzyme complex or other such changes can lead to a sharp decline in the reaction rate. So, these two examples are sufficient to show that food reactions generally conform to the Arrhenius relationship over a certain intermediate temperature range only, and deviation can occur on either side of this. Therefore, Arrhenius relationship should not be blindly applied to food system. First, depending upon the type of food, using appropriate experiments, one should find out what are those range which is following Arrhenius relationship, and then in modeling and other processes, this system or this relationship or this specific temperature range that has been experimentally tested should only be used for that is Arrhenius relationship in the food systems. And these deviations in the Arrhenius relationship, if we want to summarize, the reasons for this deviation may be because of the enzyme activity might get lost depending upon the temperature and other factors. There may be deviation in the reaction pathway which might get influenced by competing reactions. Physical system or physical state of the system may change, and one or more of the reactants may become depleted, and these all factors together or individually they influence that relationship. We see the next variable, time. It is an important factor to be considered, but the time should be considered with the rate at which the temperature changes with time. For example, during storage of a food product, it is generally desired to know that what is the capability of the food. Capability of the food means with respect to its quality, that is focus is on the time with respect to the integral of chemical and microbiological changes that occur during a specified period of time. And in this, obviously the rate becomes an important consideration. During processing, there is the time variable at which appears in the rate expression must be considered. The relationship, that is dt by dt, is important because it determines the rate at which the microorganisms are destroyed or the relative rates among the competing chemical reactions. So, it is not only the change in the temperature or it is not only the change in the time, but integral of the change in the temperature with time, at which rate, whether the temperature is changing very fast, temperature is changing slow, time is more, time is less. So, this at least rate of the change of the temperature with time is important. Okay. And in fact, this rate has also important influence on the relative concurrent reactions, right. That is this in the system, food is a complex system and various reactions takes place simultaneously. So, this change in the reaction rate might influence the reaction processes, that is the processes which are occurring constantly. For example, if you can say that a food can deteriorate both by a lipid oxidation and a non-enzymatic browning reaction, and if the product of browning reaction is antioxidant or the products which are formed as a result of browning reactions, maybe browning reactions little chemistry or more detail we will study in the other classes, and where the things will become more clear, that end product of the browning reactions may be antioxidant. So, if it is so, it becomes important to see that if the time or rate of these two reactions are overlapping with each other to cause a beneficial interaction or to cause a sufficient interaction among these. Other variable, pH, it influences the rate of many chemical and enzymatic processes. In fact, most of the enzyme they need optimum pH for their best activity. Even extreme pH values are usually required for severe inhibition of microorganisms or even growth of microorganisms, or even enzymatic reactions, their rate of the enzymatic reactions, all these are influenced by changes in the pH. The pH of the material, it may result in the acceleration of acid or base catalyzed reactions. In contrast to this are other factors, that is there are various examples like in the case of mussel or in some other foods. Even small changes in the pH can cause profound change in the quality of the food. The change of the pH of the mussel post slaughter changes its textural characteristics, sensory characteristics, even increase or decrease in the pH of the food might increase or decrease the taste of the food. So, all these things it becomes very important variable one must consider during processing, that is the pH of the material, pH of the, that is the, when we want that an enzyme reaction should proceed, and if it is the target to have a properly controlled enzyme reactions or properly controlled microbiological reactions, we must see and that the pH of the food tallies or it is in the range of the optimum pH for the maximum or best activity of that enzyme or that microorganism. Other important variable which should be considered by processors or by others who are handling food material, that in the industry or anywhere, that the composition of the product, both the raw material or ingredients which are used for the processing or manufacturing, as well as that is the end product characteristics, what are even the material which are added into the food, because it is the property or characteristics or composition of the ingredients, quality of the ingredients which actually governs or controls the quality of the end product. Composition of the raw material and composition of the finished product quality are very important. Okay, that is. We can take one or two examples of a plant system or animal system to explain this little better. That is, fruits and vegetables post harvest, the manner in which they are handled can cause increase in the sugar content. Let us see further specific example of potato. There is potato, normally it contains starch, but after its harvest, how it is stored, right, how it is handled, and if there are fluctuations in the temperature or even temperature handling conditions etc., may cause hydrolysis of the starch, and if it so happens there might be more amount of glucose or even sometimes fructose sugar. All right. So, this increased amount of reducing sugars will cause more browning during the preparation of chips, during drying steps or during frying steps etc. So, that becomes this manner in which these fruits and vegetables are handled post harvest. Okay. It will influence the change in the color or change in the various quality attributes during its drying or frying or such other operations. Similarly, in the case of muscle tissues, as I also told earlier, that after slaughtering, the manner in which these muscles are handled, the temperature during handling, other process, aerobic conditions and other conditions in the system, they influence the extent and rate of proteolysis, glycolysis etc., and which ultimately affects the texture, flavor, color and other attributes of the food material. Okay. Even the blending of raw materials, if 2 or 3 in a formulated food, in a fabricated food, where if 2 or 3 or more ingredients are used. So, the type of ingredient, the proportion in which these ingredients are blended, in the manner and how they are mixed etc., that may also cause important influence. For example, that is the rate of oxidation reaction can be accelerated or can be decelerated, increased or decreased depending upon the amount of salt present in the mixture. Similarly, in the case of, as I told you, fabricated food, even the composition of the product can be controlled by adding certain food grade approved chemicals such as acidulants, even chelating agents. Chelating agents can be added which can bind the metal ions, etc., and therefore they can stop them reacting further with the other component and bringing about changes, etc. Even antioxidants can be added to control the oxidative hydrolysis, oxidative rancidity and other reactions, okay, or even by removing the undesirable reactants from the system, one can control. For example, in this case, the removal of that is the glucose from the dehydrated egg albumin prior to its drying for conversion of egg powder becomes a very important, and in fact the color characteristic or other characteristics of the powder are improved. Otherwise, this glucose, if it is not removed, it may act with the amino acids or amines and it may result into the Billard browning reaction. The egg powder ultimately becomes brown in color. So, these becomes the reaction, that is how. Similarly, in addition to the composition of the ingredient, the composition of the environment, composition of the gaseous phase in which the material is being handled, in which the material is being processed, in which the material is being stored, that also have important influence on the properties and characteristics of the mood, and it should be properly, needless to say that it should be properly controlled. One very common example is the oxygen, that is presence or absence of oxygen may result, that is if we are storing a material which is likely to be spoiled by microorganisms and those aerobic microorganisms, then presence of oxygen might be a deterrent in that system. So, means environmental composition of the gases, etc., might influence, that is the presence or absence of the oxygen into the system, okay. And there are many situations where we want to avoid or limit the oxidation processes, and in this case, in fact, depending upon the, even if we go the instrumental method or such a third method for exclusion of the oxygen, but it becomes almost impossible to have complete exclusion, or even sometimes we do not want to have complete exclusion, because if you completely exclude oxygen, anaerobic processes may take over. So, and a small amount of oxygen might be there, but again in some materials even a small amount of oxygen, particularly in the product like coffee, etc., it might have significant influence on the characteristics or quality of the product during storage. Okay. Even early formation of a small amount of dehydroascorbic acid by the oxidation of ascorbic acid, okay, which can be even, which can take place even in at a small amount, comparatively lower amount of oxygen availability. So, it can lead to the browning reaction during storage. So, all this becomes important variable and consideration, it should be properly checked and controlled. The last but not the least variable is water activity. Okay. It is one of the most important variable that controls the reaction rate in food. Maybe in the next lecture, this water and its influence on the food stability we will study in little detail, but here I will just try to tell you that water, it is not the water, rather water activity becomes important, because it is available for the most of the enzymatic reactions, lipid oxidation, non-enzymatic browning, sucrose hydrolysis, anthocyanin degradations are all, almost all reactions are influenced by water, and most of these reactions they decrease or they proceed at a lowest possible rate over the water activity of the intermediate moisture food, that is what is 0.75 to 0.85. So, means that is water activity. To minimize these processes, water activity should be brought to 0.75 to 0.85, all right. And the reason for this is that the reduced solvent capacity of the decreased water phase, which reduces the rate of these reactions. Of course, lipid and associated secondary effects such as keratin degradation etc. are exceptions, because their rate is accelerated even at a low temperature. So, this gives an overview that how water activity influences the characteristics or quality of the food material. So, it should be properly controlled. Depending upon the different constituents present, particularly the water and other components, the world food supply is generally classified broadly into three major groups, and those are perishable, semi-perishable and non-perishable or shelf-stable. Perishable foods are those that deteriorate quickly after harvest or slaughter. Semi-perishable food contain natural inhibitors to spoilage, are those that have been given some type of mild preservation treatment and which creates greater tolerance to the environment conditions during handling and distribution. The third category, non-perishable or shelf-stable food. These are non-perishable at room temperature. Even some of these foods have been made room temperature stable by suitable processing means like canning or by drying to reduce their moisture content to a significantly low value or so. Here, in fact, in this table, I have again shown you that perishable food, they have medium to high moisture content. They can be stored for 1 to 7 days at room temperature under ambient condition etc. And the examples becomes poultry, seafoods, milk, juicy vegetables etc. On the other hand, semi-perishable foods contain medium water content. It can be stored from 1 to several weeks depending on the storage environment and other conditions. Shelf-stable foods are non-perishable foods. They have longer stability, but at room temperature they can be stored for 1 year or more, and they have low moisture content. And food grains, nuts, beans etc., they come into this category of foods. The another important aspect which we will come across to this course or in food science, it is very common term, that is food shelf life. We say that this shelf life, this food has a shelf life of 2 months, 3 months, 6 months or so. So, what does actually this shelf life means? Means it is a period, shelf life of a food is a period between the processing and retail purchase and use of a food product. And during this finite period of time, the product is in a state of satisfactory quality in terms of nutritional value, taste, appearance, flavor and its use. And this shelf life, in fact, that changes because of the many reasons, because of those factors which influence the quality and it. In commodities, particularly this plant life or other cases, that is once the material is harvested, any food material, it contains the harvested mature crop, contains a variety of oxidizable substrate and the molecular machinery which are required to perform oxidative reactions. And in all these respiration, where sugar is converted into carbon dioxide and water and energy is released and under aerobic conditions, this becomes the reaction of major concern. And in fact, that the mechanism of these respiration reactions becomes essentially the same, whether it is fruit or vegetable or in other plant and animal life. And of course, the biological oxidations involve a number of metabolic pathways in which synthetic degradative reactions are interdependent, and in most of the cases we try to control these reactions, that is the respiration and other oxidation reactions etc., to control the shelf life of the food material. So, in the next 2-3 slides, in fact, I have already much discussed about this aspect, but this again gives you a little inner view of that how that is the plant materials containing, that is the carbohydrate rich plant foods, what are the major interactions, major chemical changes among different components, all right, that is during processing and storage, and which ultimately influence the quality of the food, textural quality, sensory quality, color, flavor, nutritional value are minded. These are the same set of the reaction which also might result into the development of certain toxic components. See, if you have a clear cut idea about the department, because the carbohydrate is a very big term, it might include different polysaccharide, disaccharide, oligosaccharide, monosaccharide etc., and these depending upon the system constant, that is what are the temperature, pressure, heat, acid, light, various factors. So, when you are processing, even it may come in contact with the protein, it may come in contact with the lipid etc. So, there may be different interactions among them, interaction with the environmental factors, interaction with the processing factors, and this ultimately results into the decomposition, degradation or other change in these components and various new carbonyl compounds etc., like furfural, there may be caramelization or pyrolysis, all right, even there may be Maillard or sticker degradation reactions, and ultimately it influences the quality of the most of the product. Similarly, if we take a protein rich food, here also again proteins are polypeptide, then these polypeptide can be broken down into super and dipeptide, tetrapeptides etc., are peptides, and these again in the similar like we have discussed in the case of carbohydrate, similar here also it may get in contact with acid or certain enzymes, water, even under anaerobic condition etc., and then there will be acid, severe acid, moderate acid or other reactions, and ultimately again it may get involved into different types of cross linking reactions, oxidation reactions, Maillard reaction etc., and various new compounds get formed and the product characteristics. So, this gives you there is the major chemical reactions which take place during in the protein constituents and it interaction with other components of the food. Similarly, the lipids, here also almost similar explanation can be, that is the lipids, they may come across various, that is factors like enzyme, temperature, heat, irradiation or other air and all those things, and they may undergo different, which you can say that there may be oxidation, there may be interaction with the protein, there may be formation of that is hydroperoxides etc., and all these they may interact with these hydroperoxide, may interact with protein, with other components, and then ultimately again various carbonyl compounds and other compounds may be formed and which influence the quality of the food material. This slide shows you again that is interaction, that is when during handling or storage, the interaction between major components of the food material, and the major component as you could see it is the polysaccharide, it is fat and proteins. These polysaccharide again depending upon the factors available, they may be broken into various components. Polysaccharide may be pectic substances, celluloses, starches, they may be decomposed into smaller like monosaccharide etc., like glucose or galacturonic acid, and these they may enter to the either glycolytic cycle or pentose phosphate pathways. Finally, it converts into pyruvic acid, and at this stage even the fat, that is this can get decomposed to glycerol and fatty acid. The glycerol comes here and they enter to the TCA cycle, tricarboxylic acid cycle. Similarly, proteins, they are composed of different amino acids, and these they can be under, depending upon the different conditions prevailing, they can be broken down, and different amino acids, they enter at a different stage, like alanine, cysteine and serine, they enter at the pyruvic acid, whereas the histidine, proline, arginine, they enter at a later stage in the TCA cycle. Ultimately, the series of complex reactions occurs, and then these reactions further in the oxidation, they get oxidized, and the energy is released from the system, and other components are formed. So, this energy release process, another process. So, by this, well, ladies and gentlemen, then we have had a good overview of the how different factors during processing, handling, storage, etc. of the food can influence, can interact among themselves, can interact with other factors, processing factor, food factor, environmental factor, etc. and they might bring about changes in the constituents of the food, characteristics of the food and quality of the food. These changes might be towards the betterment of the food, these changes might be towards the spoilage of the food, and this because these are the same type of organic or carbon reactions, etc., which also take place when the food, the desirable characteristics of the food are developed, which we saw in the earlier class. So, it becomes an important consideration, that is, when we are handling the food, when we are processing the food, we should meticulously, properly control these factors, so that we get desired result in our product. Thank you.